Good evening to all the students of ICSE and CBSE board. Today we are going to start a very interesting chapter, that is life processes. In that we are going to deal with the chapter photosynthesis, which is from ICSE syllabus. So first let us look into what are the life processes. For life to take place in all the living organisms, starting from unicellular to multicellular organisms, all the processes have to occur inside the body. For example, nutrition, metabolism, respiration, transportation, excretion and reproduction. So, all these processes which take place in a coordinated manner, and then all the organ systems of the organism will function in a coordinated manner or in a coordinated fashion. So, look into the theory once. All living organisms are made up of structural and functional unit called cells. The basic life will start from atom, but then cell is addressed as the structural and functional unit of life. Cell is addressed as the structural and functional unit of life, because a single cell whose components include a cell membrane, cytoplasm and genetic material. A single cell will have a genetic material, whether it is surrounded by nuclear membrane or not, and then it has nucleoplasm or cytoplasm. Nucleoplasm will be there inside the nucleus, and then cytoplasm inside the cell, and then cell membrane. Right? These are the three components of a cell. Such cell is structurally and functionally independent. Cells are made up of protoplasm. Protoplasm is nothing but cytoplasm along with the genetic material is addressed as protoplasm. To sustain the life processes, various biochemical reactions take place. All the biochemical reactions which occur in the coordination manner will take place in the life processes. Some of the important processes required for the life include, what are those processes? Nutrition, metabolism, respiration, reproduction, excretion and transportation. These are addressed as what ma? Life processes. In that, first we are going to speak on nutrition. When nutrition is considered, any living organism which is taking the nutrients or food material to run the metabolic system or to run all the processes, metabolic processes in the organism, is addressed as what? Nutrition. There are different modes of nutrition. If you see, different modes of nutrition, it can be autotrophic or heterotrophic. Let us put it in a flow chart. Nutrition is of two types. Autotrophic mode of nutrition and heterotrophic mode of nutrition. So here if you observe, autotrophic mode of nutrition can be operated in two methods. One, it is photosynthetic autotrophs. Second one will be chemosynthetic autotrophs. Photosynthetic autotrophs, where they take the energy in the form of sunlight. The energy in the form of sunlight, and electron donor will be H2O or H2S here. Photosynthetic autotrophs are again of two types based on the requirement of the electron donor. Photosynthetic autotrophs are of two types based on the electron donors. One conducts oxygenic photosynthesis. Second type is anoxygenic photosynthesis. In oxygenic photosynthesis, the electron donor is H2O. H2O is the electron donor. In anoxygenic photosynthesis, the electron donor will be H2S. Clear, right? And the energy is taken from sunlight. Sunlight is the ultimate source of energy. And then chemosynthetic autotrophs will consume the energy to prepare the solid food material or complex molecules. What is autotrophic mode of nutrition? Autotrophic mode of nutrition is nothing but preparation or synthesis of food materials which takes place in photosynthetic or chemosynthetic bacteria, small or simple algal cells, and all the higher plants. They take place in autotrophic bacteria, algal cells, and all the higher plants. If the energy which is required for the preparation of food material is taken from sunlight and the electron donor is either water or H2S for oxygenic and anoxygenic photosynthesis, then the mode of autotrophic nutrition addressed as what? Photosynthetic autotrophs. If at all the energy which is taken is ATP, the energy which is consumed is ATP, which is chemical form of energy, and the electron donors are inorganic elements, like nitrogen, iron, sulfur, etc. Chemosynthetic autotrophs will help in recycling the nutrients as well. So, autotrophic mode of nutrition is operated in higher plants. Along with that, you can also observe in algal cells and autotrophic bacteria. Autotrophic nutrition is operated in two methods based on the energy it is consuming and based on the electron donors. Here the energy consumed is sunlight, ultimate source of energy, and the electron donors include water and H2S. Whereas in chemosynthetic autotrophs, the energy is ATP, which is chemical form of energy, and this energy is being taken after oxidation of food materials which is there. Okay? And then the inorganic elements like nitrogen, ferrous, sulfur will be there for chemosynthetic autotrophs. Next, heterotrophic mode of nutrition is operated in herbivores and carnivores, which are primary and secondary consumers. And heterotrophic nutrition is done in three methods. First is saprophytic mode of nutrition. Second, symbiotic method. Third, parasitic. Saprophytic mode of nutrition is mostly observed in fungal species or fungi, where the organism will be releasing out the enzymes. For example, this is the organism. In close proximity to the organism, there are food materials present, complex food materials like debris. Most of the decomposers will rely on saprophytic mode of nutrition. So, they will release the complex, they will release the enzymes which will digest the complex substances into the simpler substances, and later these simpler substances are assimilated into the organism. Okay? This is saprophytic mode of nutrition, which is seen mostly in the fungi or decomposers. Symbiotic mode of nutrition, where the organisms which are involved in symbiotic association will be benefited in the dual ends. Best example I can give you is Rhizobium bacteria, Rhizobium leguminosarum. It will help in nitrogen fixation in the legume roots, whereas legume roots will provide shelter to the bacteria likewise. And you can also take the example of lichens, which is the association of algae and fungi, where both the organisms are getting benefited. Algal component is known as phycobiont, fungal component is known as mycobiont. Okay? Both will get benefited in their association, where algae will provide nutrition, fungi will provide shelter. Last mode of heterotrophic nutrition is parasitic method, okay, wherein the organisms will infect the host. Okay, it will either cause death of the host or infection to the host, and it will rely on the host for the nutrition completely. Okay, so this is autotrophic and heterotrophic mode of nutrition, right? I hope the types of autotrophic and heterotrophic mode of nutrition are clear to you. And now we will start on discussing about photosynthesis. Okay. So what are life processes basically? At the conclusion, what are we learning? All the processes which are involved to run the metabolic reactions inside a living organism are life processes, like digestion, circulation, respiration, reproduction, excretion. All these processes occur in a coordinated manner, right, either in the plant body or in the animal body. And now we are going to discuss on the most important concept, that is photosynthesis. Because of this process, all the living organisms present on the earth are getting the nutrition. So photosynthesis is provided for all the organisms. This photosynthesis has got few synonyms. Photosynthesis is addressed as physicochemical process. Physicochemical process. This is also known as anabolic process. This is also known as anabolic process. This is also addressed as oxidoreductive reaction or oxidoreductive process, right? It is known as physicochemical process because the physical form of energy, that is light, is absorbed in the form of photons, and it is converted to chemical form of energy inside the stroma of chloroplast. That chemical form of energy is ATP. So the physical form of energy is converted to chemical form of energy. This is what physicochemical process. Just remember, physical form of energy is converted to chemical form of energy. Which is photons is converted to chemical form of energy, that is ATP. Next, anabolic process. Any process which involves the conversion of simpler molecules into complex molecules is known as what? Anabolic process, which is one of the step of metabolism. Second step of metabolism involves catabolism, where complex molecules are destructed to simpler molecules. So photosynthesis is said to be anabolic process because the simpler molecules like carbon dioxide and water, in the presence of light and chlorophyll, they are converted to the complex molecule, that is glucose. Okay, they are converted to the complex molecule, that is glucose, right? And last, it is said as oxidoreductive process. This we will understand clearly when we look into the standard equation of photosynthesis. Let's now look into the standard equation of photosynthesis, wherein 6 CO2 plus 12 H2O in the presence of sunlight and chlorophyll. Okay, this forms glucose C6H12O6 plus 6O2 plus 6O2. Okay, this is the standard equation of photosynthesis. In the products, what is the main product? Glucose is the main product or primary product of photosynthesis. Glucose is the primary product of photosynthesis, and oxygen is evolved as byproduct. Oxygen is evolved as byproduct. So now here if you look into the reaction, what are the raw materials of photosynthesis? Carbon dioxide, water. What are the products of photosynthesis? Glucose is the main or primary product and oxygen is the byproduct. If you clearly look into the reaction, carbon dioxide, which is the substrate, when it has turned to the primary product, it got reduced from CO2 to glucose. It got reduced or not? Addition of hydrogen group. Next, water molecule, if you see, it got oxidized to oxygen. So here oxidation and reduction both are happening. Oxidation and reduction both occur in a single reaction. So this is addressed as oxidoreductive process or oxidoreductive reaction. Photosynthesis is very important for two main reasons. It holds two main reasons. It is a primary source of food on earth and it is responsible for the release of oxygen into atmosphere on which most of the living organisms survive. Okay, on which most of the living organisms survive. This is all about what? Introduction of photosynthesis. You have to remember, photosynthesis is a physico-chemical process in that what is oxidizing and what is reducing. Carbon dioxide is getting reduced and water molecule is getting oxidized, and it is said to be anabolic process. Anabolic process because simpler molecules are converted to complex molecules. And then physico-chemical process because physical form of energy, that is photons, is converted to chemical form of energy, that is ATP. Now, okay, we have understood the standard equation of photosynthesis. Why is it called as anabolic process, oxidoreductive process, physico-chemical process? But then who verified this? The standard equation of photosynthesis is proposed after series of experiments. Okay, so the first experiment, if you see, that is variegated leaf experiment. In simple terms, you will address it as what? Starch test. So for this test, what do we need? Take a potted plant. Take a potted plant. Take a potted plant, place it in dark for 3 days. Place it in dark for 3 days, and then again provide it with sunlight after 3 days of dark. The main purpose of keeping the potted plant in dark is to destarch it. To destarch it. What does this term mean? Destarching. All the starch which is present in the leaf should be utilized by the plant to run its machinery. All the starch present in the leaves. Starch is the stored food material in a plant cell. All the starch present in the leaf should be utilized by its machinery. So for destarching, what are we doing? We are placing it in dark. If it is in dark room, sunlight will not be available. It cannot prepare the food material. So it will rely on the starch which is present. So to destarch it, we are placing it in dark. After that, we will provide 6 hours of sunlight. To make its metabolism active. Whenever you are hungry, too much hungry, food is provided at that time, so you will eat it very in a higher frequency, right? Same we are applying to the plant here. After 3 days of keeping it in dark room, we are again putting it under sunlight for just 6 hours to activate its metabolism. And then you take the leaf, boil it, boiling to arrest the cell, to arrest cell. Cell arrest will happen here. Then to remove the chlorophyll present in the leaf, okay, you have to place it in alcohol. Here chlorophyll also will be removed when you are boiling it, and the cellular machinery will get arrested. All the cell metabolism will be stopped. And then you have to place it in alcohol. Chlorophyll will be lost, and then you can see some regions of the leaf will be dark and some regions of the leaf will be light. This dark because what? 6 hours you placed it under sunlight, na? So some part of the leaf will be conducting the photosynthesis and preparing the starch again. So some parts of the leaf will be dark and some parts of the leaf will be light. Now this leaf should be dipped under iodine solution. It should be dipped under iodine solution. After dipping in the iodine solution, you can observe only the green parts of the leaf. The green parts of the leaf hold starch. The rest will be pale in color like this. Only some part of the leaf will be green because of the action of, because of the interaction with iodine. It forms what complex? Starch iodine complex. Okay, it forms starch iodine complex. And then it turns into pale color. Rest part wherever starch is not present, it will remain pale itself. Okay. So once again, this experiment is known as variegated leaf experiment. A variegated leaf is a leaf which is not completely green in color. Okay. Some parts will be of yellow and some parts will be green. Okay. So photosynthesis occurs only in the green parts of the leaves in the presence of sunlight. To confirm that statement, this variegated leaf experiment is done. Okay. Wherein first a potted plant kept in dark for 3 hours. Immediately after destarching it, we are providing sunlight only for 6 hours, and then the leaf will be boiled. Boiling of the leaf for what purpose? Arresting the cell. Cellular arrest takes place. Placing the leaf in the alcohol to remove the chlorophyll, and then dipping it in the starch, dipping it in the iodine solution. Only the green parts will turn what? It will turn blue-black. The rest part will remain pale. This will indicate what? Starch iodine complex. Got it? So this is about variegated leaf experiment. So with this they proved what? Starch is the food which is prepared by the plant cell. Glucose is the product of photosynthesis. What are the raw materials? Carbon dioxide and water. Who proposed that carbon dioxide is essential for the process of photosynthesis? Carbon dioxide is essential for the process of photosynthesis is proven by whom? Moll's half-leaf experiment. Symbol experiment. A potted plant which is placed under sunlight only, half of the leaf should be dipped under KOH, potassium hydroxide solution, half of the leaf. Okay, like you can, we have done this experiment with honey bottles, where the leaf will be fitting into it. Only half of the leaf will be sent into the honey bottle. That bottle consists of what? Potassium hydroxide solution. Or if you have the exact stopper also, you can use that stopper and you can place the leaf. So this potassium hydroxide solution will completely absorb carbon dioxide present in the leaf, this half leaf. So when carbon dioxide is completely absorbed by KOH without CO2, it will not perform photosynthesis. Like that, it is placed for some period of time. After that, the leaf which is kept into the KOH solution, that leaf is plugged and performed with what? Starch test. starch test. After starch test, it is proven that only half of the leaf which is exposed to sunlight has abundant amount of carbon dioxide and it is performing the photosynthesis. And the leaf which is placed into the KOH solution, it does not have any carbon dioxide, it does not perform any photosynthesis, so it is not responding to what? Iodine. Like that you will see half of blue black color and half of pale color after starch test is done, okay, when it is placed in iodine. So with this moles half leaf experiment, he has proved that carbon dioxide is essential for the process of photosynthesis. And then the experiment is Joseph Priestley experiment, which is on oxygen discovery, okay. This is nowhere related to the photosynthesis. He has discovered oxygen and there is some gaseous exchange between the plants and animals is proved by him, okay. What did he do? First, in the first phase, he has placed a mice with a candle, closed it with a bell jar without any gaseous exchange. Later what did he see? After some time the mice dies and candle puts off, okay. He has repeated the experiment by placing a mint plant into the bell jar. After some time he can see the candle is still lightening, mint is fresh, mice is alive, whereby he proved that the air which is released out by the plant is taken by the animal and the air which is released out by the animal is consumed by plant. So there is the gaseous exchange which is taking place. All around 1770 he has performed this experiment. He has discovered oxygen in the year 1774. He has discovered oxygen in the year 1774. Clear Nuttall here? And then Jan Ingenhousz experiment. He is the scientist who proposed that photosynthesis takes place during sunlight and in the present, and it will release oxygen as a byproduct. Photosynthesis occurs in sunlight and it releases oxygen as byproduct. It is proved by Jan Ingenhousz experiment, wherein he has done the experiment on hydrilla, so it is also popularly addressed as hydrilla experiment. Hydrilla experiment. Right. So what is this hydrilla experiment? In green plants, sunlight is necessary for the process of oxygen production. What did he do? Very simple, ma'am. A beaker containing water and a tistil funnel is taken. Tistil funnel which will be like this, right? So into that tistil funnel he has placed what? Hydrilla plant. He has inverted the tistil funnel and placed it in the beaker containing water. Over this funnel he has placed what? Test tube and closed this completely. Two such setups were prepared. One is kept under the sunlight and the other is kept in dark. Got it? What did he do? A beaker containing water and a tistil funnel is taken into which the hydrilla plant is placed. He has inverted that, kept it in the beaker and closed this funnel with what? Test tube. Two such setups were made. One is kept under sunlight and the other is kept under dark. Okay. After certain period of time, he has observed that the setup which is placed under the sunlight is showing some bubbles in the test tube. It is showing the bubbles in the test tube. Okay. When this is observed, for what gas it is releasing? When a burning matchstick is placed into that test tube, open flame, it is burning more brightly. It is the indication of what gas? Okay. So oxygen is released only in that setup which is placed under sunlight. Okay. So who is that scientist who proved? Yes, this is important for the exam, ma. Listen carefully. So who proved this experiment? Jan Ingenhousz. What are the points he has proved with hydrilla experiment? That sunlight and oxygen are essential for photosynthesis. Are essential for photosynthesis. Okay. Next. Yeah, sunlight is essential and oxygen is released as byproduct for photosynthesis. Accepted? Thank you. Next, Julius von Sachs. He is the scientist who said that glucose is the product of photosynthesis. Same by the variegated leaf experiment. We have discussed first, no, that only. This is there for your NEET also, so listen, will be helpful. So what are the discoveries we have learned? First we have learned photosynthesis is oxidoreductive process, physico-chemical process and anabolic process. When you are saying it physico-chemical process, you should know the meaning. Physical form of energy, photons, are converting to chemical form of energy, ATP. Oxidoreductive process, you should know the meaning, which is getting oxidized. Which is getting oxidized? H2O is oxidized. Which is getting reduced? CO2 is getting reduced. And third is what? Jan Ingenhousz experiment. That is hydrilla experiment. Bell-jar experiment is clear, no? Oxygen discovery, some gaseous exchange between plant and animals. And then carbon dioxide is essential for photosynthesis said by Mohs half-leaf experiment. Jan Ingenhousz, who did hydrilla experiment, proved that sunlight and oxygen are essential for photosynthesis. Clear not in here? Clear? Next, chlorophyll or structure of chloroplast we will learn. Structure of chloroplast. Okay, chloroplast is a double membrane bound organelle. The space between outer and inner membrane, is addressed as periplastidial space. Periplastidial space. And inside the matrix of the chloroplast is filled with a fluid space that is known as stroma. Stroma is the matrix of the chloroplast. And then inside the chloroplast there are some membranous sac-like structures. These membranous sac-like structures are called as what? Thylakoids. Thylakoid also possesses a membrane over it, okay, that is also referred as third membrane system of chloroplast. Okay, this is also referred as third membrane system. Of chloroplast. And this thylakoid membrane is the site where it contains chlorophyll pigments. What is the site of chlorophyll pigments? Thylakoid membrane or thylakoid lamellae. Right? And this single sac is known as thylakoid, membrane is known as thylakoid membrane, stalk of this sac-like structures is known as what? Grana. Grana is stalk of thylakoids. One grana is connected to other grana by what? Stroma lamellae. And stroma of the chloroplast, which is the matrix, right? Stroma consists of what? Stroma consists of double-stranded circular DNA. It consists of double-stranded circular DNA, for which it is addressed as semi-autonomous cell organelle. It has double-stranded circular DNA. It has starch granules as storage bodies. It has lipid droplets as well. Lipid droplets. Clear? This is all about the organelle chloroplast, which is semi-autonomous cell organelle or self-duplicating cell organelle. Clear, right? It also has 70S ribosomes. Very good. It has 70S ribosomes. So, both the features, double-stranded circular DNA and 70S ribosomes, are in similarity with what? Prokaryotic cell. So, there is a theory called as endosymbiotic theory. Endosymbiotic theory, which says that chloroplast and mitochondria are evolved from prokaryotic cells. Long back they are also the individual prokaryotic cells. So, both chloroplast and mitochondria can generate their own ATP. Okay. In due course of time, the eukaryotes have engulfed the chloroplast and mitochondria. So, they have become the part and parcel of eukaryotic cell. Got it right? So, what are the two similar features with the prokaryotic cell? Double-stranded DNA and 70S ribosomes. Clear, right? This is about the structure of chloroplast. About the steps of photosynthesis, sites of the reactions, we will discuss in the next class. Before ending up the session, let us solve few PYQs on the discoveries that we have learnt. Yeah, first question. These are the questions, MCQs from your ICSE text. The production of starch and not glucose is often used as a measure of photosynthesis in leaves, because starch is the immediate product of photosynthesis. Wrong. What is the immediate product of photosynthesis? Glucose. Glucose formed in photosynthesis soon gets converted to what? Starch. Correct. Starch is soluble in water. Wrong. Starch is insoluble in water. Starch cannot be tested. No, starch can be tested with what? Iodine. Okay. Sugar can be tested. Wrong. Okay. So, the right answer is option B. Glucose formed in photosynthesis soon gets converted to what? Starch. And then this we can do after learning light reactions. Yeah. Chlorophyll in leaf is required for what? Chlorophyll in leaf is required for breaking down water into hydrogen and oxygen? No. Emitting green light, trapping light energy, storing starch in the leaves. Trapping light energy. Yeah. Which one chemical reaction occurs during photosynthesis? Last question of the session. Which one chemical reaction occurs during photosynthesis? Which one? Carbon dioxide is reduced and water is oxidized. That is why it is known as what, ma? Oxidoreductive process. Oxidoreductive process. Yeah. Thank you all, and in the next class we will look into the steps, phases of photosynthesis, and we will solve more questions from the syllabus. Thank you.